Hello, hello, hello. So I'm glad to see that some of you are here. So some of you already finished all the lab work, which is amazing. So now all you have left is lecture. Okay, so I have a few announcements to make. First, final lecture exam a week from today. So today is the 9th. So a week from today, notice the time, 1:45. So you must come to, you can hear me, right? I forgot to ask.
Yes,
You
we
can
can.
and see
Yes.
me, right? Okay, so please don't forget to come to the assembly hall. It's in person, the exam, as usual. So this is our third lecture exam. So it starts at 1:45. So please arrive a little bit earlier because we're going to have attendance. We still haven't worked out how the attendance is going to be, but I will let you know 2 or 3 days before the exam. We're trying to have these iPads that you can just show the scan, the either your phone or the physical ID. We're still not sure how this is going to work, but I will let you know. So 1:45 and the exam will end at 2:45. There will be no time or date changes. Please understand, I wish I could have one date, a different date for each of you, but I can't. There's so many students. It takes a long time to make the exam. Also, I like to have the same exam for everybody. I think that that's fair. So, and you've known from day one of class, back in August, August 26th, which was our first class, I announced the day of the exam and the time. So if you have some conflict, please ask the other professors to make some change. Okay? So um, the day of the exam and time is the same. It doesn't change. Now, after 2:45, some students need to take a makeup. I'm in trouble here. So the makeups will start right after the final. So that means that for some students, you'll have to download two exams, the final exam, which you will take first, and then the makeup exam, which will be exam one, two, one, or two. So those students send me an email confirming that they have to take a makeup only those students will have access to the makeup, not the whole class. Only those students that have confirmed by email to me that they will be taking the exam, only those students will have access. I don't know why. Oh, will have access to the makeup. A very, very important announcement. By now, you should have all your grades for lecture and lab 1, 2, and 3. Some students, the grade for lecture exam 2 was not visible. So if you don't see the grade for lecture exam 2, and you know you took the exam and you uploaded and submitted the exam, please send me an email. So right now, after today's lecture, please go to Brightspace and confirm that all your grades for lecture and lab exam 1, 2, and 3 are there. There is no attendance for lecture on Brightspace. That is absolutely correct, Kevin, because the attendance will be in the assembly hall. So you will swipe before you go into the assembly hall. Each student will swipe their ID, and when they get out, they swipe again. So there's no grades, nothing. It's to make sure that only the students that are in the assembly hall will take the exam. If we don't have attendance for a student, then and the student takes the exam, that grade will not count. So this is to make sure that only the students that are in the assembly hall and that are being proctored, those are the only grades that will count. Okay? So that's the reason for this. But please make sure that your lecture and lab exams are all correct, and also the attendance. Some of you will start having the grades for lab exam 4. I've asked the lab instructors to post the exam grades 24 hours after your lab exam. So make sure if you have any problems with your grades, now is the time to contact your instructor, your lab instructor, or me. Yes, if there's a discrepancy only in the lecture, email me. If you have a discrepancy in the lab, email your lab instructor. Okay, Mohammed, I just explained how am I going to take attendance for the lecture exam. There's going to be a system, an iPad, where a student, they will be at the doors of the assembly hall. And when a student goes in, you scan your ID. And that's how we will know which students are in the assembly hall. Once you are in the assembly hall, you cannot leave the assembly hall. Okay? So these discrepancies, now is the time, because after the final lecture exam, there's not going to be any changes in grade. That's it. Your grade is final. So please make sure that everything is okay now. You have time from now until the final. So look at number 6. All grades will be considered final after Wednesday the 17th. Yes, arrive early, not at 1 p.m. There won't be anyone there because there's classes. There are other classes that are taking the exams. So arrive like at 1:30, 1:15, something like that. Okay? Everything is going to be fine. Okay. Next announcement. So next class, which is next Thursday, will be our last lecture. So what I'm going to do is I'm going to have a review. So I'm going to make up lots of questions similar to the ones that will be on the exam, not the same ones, of course, but I'll try to have the same style. And that's what we will do in the next class on Thursday. It will be just review. But you have to watch this movie. So this movie is available online. You can just click on this link. It's about 55 minutes film, and it's very interesting. It's about Parkinson's disease and frozen addicts. It's like a Sherlock Holmes detective movie for a clinical aspect. So there will be two questions related to the movie. So please watch it. I think you will find it very interesting. I know these are stressful times because you have exams and so on, but you can take 55 minutes off, have a bucket of popcorn, sit down and watch the movie. I think you will enjoy it.
This is the same movie you spoke about last time, right?
Yes, exactly.
Okay.
What if we only have a digital ID on your phone? As long as it works. So for you to come in to Hunter, you also scan your phone ID. So it should work. And, the proctors will have a list. So each proctor is going to be in charge of a group of students.
Um,
So everything will be very well organized. Don't worry.
Professor, just in case if we lose our ID, can we give our ID number and they can pull us up?
No, we have to see a picture of your ID. You cannot just give the ID number. No, you have to show your ID. You have to show the physical ID or have it on your phone with a picture. Okay? Oh, okay. Thank you for telling me the waiting room. Okay. So, okay, you have digital IDs, but those digital IDs, don't they have a picture, your picture on it? There has to be a way that we can, yes, so that's fine. That's not a problem. Okay. Okay, don't forget about the movie. Okay. So, I wish we didn't have to do this, but unfortunately we do because some students try to take, these students that try to cheat in this exam should not be should not be have the privilege of being in health sciences and helping citizens that are in need, citizens that are sick and that need help. These students that try to cheat should not have permission to go into this field. It's unfortunate that some students do this. There are not that many, which is good. Okay, so let's go today. We're going to discuss the autonomic nervous system. So we spend a lot of time on the central nervous system, the spinal cord, the brain, all the different parts of the brain and their functions and so on. So today, one lecture dedicated to the autonomic nervous system. So the autonomic means that it is independent of voluntary control. So the central nervous system is not required for this nervous system to function. And what does it regulate? And we are lucky that we don't have to voluntarily regulate the function of our organs. So this autonomic nervous system, its function is to regulate the smooth and cardiac muscles and the glands. The central nervous system, its function is to regulate skeletal muscle and also the intellect. So, so there's no conscious control of the autonomic nervous system, but So that means that an autonomic nervous system has a basic function that is independent of the central nervous system. But that doesn't mean that the central nervous system can have some regulatory activity on the autonomic nervous system. So the hypothalamus, pons, and medulla, they all send input into the autonomic nervous system. So the autonomic nervous system is independent. It can work independently of these areas of the brain. But these areas of the brain can have input onto the function of the autonomic nervous system. So the hypothalamus, pons, medulla, and the limbic system, which is emotion, and other regions of the cerebrum. So the autonomic nervous system is independent, can independently function, but the CNS can still have some regulatory function on it. So let's compare the somatic and the autonomic nervous system. So the somatic nervous system is the one that regulates skeletal muscle, so voluntary movement of our body and so on. So what is the input? Where are the receptors for the somatic nervous system versus the autonomic nervous system? So the receptors are the special and somatic senses. So what does that mean? Hearing, vision, touch, and so on. Proprioceptors are those located in the muscles and tendons. We already described these. And the autonomic system, those receptors are also coming from eyes and so on, and special touch, temperature, and so on, but mostly are located in our organs or visceral. The control of the somatic nervous system is voluntary, is conscious. We can voluntarily control the movement of the skeletal muscle. The autonomic nervous system is under involuntary control. How about the motor neurons? So both systems, they only have one sensory neuron. What does that mean? That means that the action potentials are conducted from the receptors towards the CNS through one sensory neuron. How about now the output, the information coming from the from the CNS out to the effectors? So in the somatic nervous system, we just have one motor neuron, and it releases acetylcholine. So it's a cholinergic neuron that is always excitatory. It's always excitatory and carries the action potential to the skeletal muscle, which are the effectors. The autonomic nervous system, now the motor neurons, there are two. One is preganglionic and the other one is postganglionic. And remember, the synapse, the preganglionic synapse is on the postganglionic at the ganglion. What is a ganglion? A ganglion is a group of cell bodies of neurons that are outside the CNS. If it would be in a group of neurons, of neuronal cell bodies that is in the white matter of the CNS, those are known as nuclei. But neuronal cell bodies outside the CNS, they are located in ganglia. So the preganglionic neurons in the autonomic nervous system, they are cholinergic. They release acetylcholine. The postganglionic, they can be cholinergic or adrenergic. That means the neurotransmitter can be acetylcholine or norepinephrine. So what are the effectors in the ANS? The heart, smooth muscle, and glands. And the response can be excitatory or inhibitory. It depends on the neurotransmitter and also on the receptors. Now, the autonomic nervous system has two motor divisions, sympathetic and parasympathetic. So this refers only to the motor part of the autonomic nervous system. So the sympathetic is activated under emergency situations. So flight or fight. That's the sympathetic. Now, under homeostatic conditions to maintain homeostasis, that's the parasympathetic nervous system, that the function of it is to maintain homeostasis. So you have these two motor divisions in the autonomic nervous system. Okay, so now let's look at the neurons, right? Which I have been describing. So here in blue, you have the sensory neurons that bring up here, this bar represents the central nervous system, so brain and spinal cord. Here it represents the peripheral nervous system. So sensory neurons for CNS and ANS, there's only one. And these sensory neurons have the cell bodies outside the CNS in ganglia. So one. It doesn't matter if it's voluntary CNS or involuntary ANS. How about motor neurons, where you have the action potentials being carried from the CNS out to the periphery? So in the CNS, voluntary, you just have one motor neuron that conducts the impulse to the skeletal muscles, which are the effectors. Now, in the ANS, the autonomic nervous system, you have two different arrangements. You have in the sympathetic nervous system and parasympathetic, there's two motor neurons. A preganglionic, which is short in the sympathetic nervous system and long in the parasympathetic. And then you have the postganglionic, which is long in the sympathetic and very short in the parasympathetic. So you might ask, why? Why is it that for the autonomic nervous system, why do you need two motor neurons? Why couldn't you have an arrangement where you would have just one motor neuron? Well, one of the advantages of this is to save space. So all these ganglia are outside the CNS. Now, you know that you have many viscera, you have the autonomic nervous system controls a lot of the different organs in our body. Here, you just have the skeletal muscles. Here, you have all the organs in our body, smooth muscle, cardiac muscle, glands. So there's a lot of control, a lot of effectors. So it's a very good strategy to push those ganglia outside of the CNS. And that's why now we have two pre- and postganglionic neurons to conserve space. Also, one preganglionic neuron can innervate many postganglionic neurons. So you're pushing this all outside. It would be very, very crowded if all this control of the autonomic nervous system would be located in the spinal cord, in the central nervous system. Okay, so now in terms of myelination and receptors and neurotransmitters. Okay, so on this side, you have the parasympathetic nervous system. In the middle, blue is sympathetic and pink is somatic. So let's start by the somatic, which is the simplest, where you have just one motor neuron and it releases acetylcholine, and the receptors on the skeletal muscle are nicotinic receptors, and they're all excitatory. Okay, now let's move to the parasympathetic, which is the second less complex one. So you have one preganglionic neuron which is very long, and then, which is not shown here, but then you have a short postganglionic neuron. So the preganglionic neuron, releases acetylcholine, and the receptors on the at the synapse on the postganglionic neuron, they're nicotinic, just like here, nicotinic. Now, the postganglionic neuron is also a cholinergic neuron. So the receptors on the organs, smooth muscle, cardiac muscle, and so on, they're all muscarinic. So nicotinic and muscarinic. And I will review what does this mean. So the nicotinic is excitatory. The muscarinic can be excitatory or inhibitory. Okay, now the sympathetic. Sympathetic, you have, it's more complex. So here you have the preganglionic neurons. They all secrete acetylcholine. So you see that all the preganglionic neurons secrete acetylcholine, and the receptors on the postganglionic or nicotinic. Another thing that these preganglionic neurons have in common is that they are myelinated. So you want a quick response coming from the spinal cord. So they're all myelinated. The postganglionic neurons, now if we focus on the sympathetic, the postganglionic neurons, some are cholinergic. They release acetylcholine, so on sweat glands, and the receptors are muscarinic. Other postganglionic neurons are adrenergic. So the neurotransmitter is norepinephrine or noradrenaline. And the receptors are noradrenergic receptors. And the adrenal gland, which is part of the sympathetic nervous system, the adrenal medulla, has receptors for acetylcholine, and it releases epinephrine that can act on, for example, the heart. So you see here that the situation in the sympathetic nervous system of the postganglionic neurons is complex. You can have norepinephrine, you can have epinephrine, and you can have acetylcholine. Now, all these postganglionic neurons are unmyelinated. What does that mean? That means that there's no melanin here. And why is that? Well, first of all, myelin takes a lot of ATP. Energetically, it's very expensive. So, okay, it allows for the action potential to travel very quickly. But think about all those oligodendrocytes and the Schwann cells. All that apparatus is very energy consuming. So having these not being myelinated conserves energy. And these impulses that are conducted on the postganglionic neurons, you don't need them to be so fast. They can be slower. So not having myelin is not so important. Okay, so here you have, again, the sensory neurons. Some of the sensory neurons are myelinated, others are not. So now we are focusing on the autonomic nervous system. So let's see here the whole situation, the whole architecture. So you have the hypothalamus, pons, and medulla that confer signals to the ANS. So they control the ANS. So you see here the neurons that are myelinated. Okay, now here's the sensory neuron. Can be myelinated on or not myelinated. Now we're going to the motor neurons. Here you have the motor neurons. So for the parasympathetic, so both preganglionic neurons, I'm pointing to them, the two preganglionic neurons are myelinated. So for the parasympathetic, they're long. For the sympathetic, they're short. So they synapse at the ganglion. And now, oh, wait a minute. This we're still in the central nervous system. Why is myelin not as important for postganglionic neurons? For the sympathetic nervous system, flight or fight, wouldn't it be faster response, be better for survival? Emily, very good comment. Now, think about the sympathetic nervous system. So the message conducted by the sympathetic nervous system is broad. So when there's an emergency situation, there's several organs that come into place. You have, for example, salivary glands. You have heart. You have sweat. All these are going to be controlled by the sympathetic nervous system when there is an emergency situation. But the length of time at which the sympathetic nervous system is going to be active under this emergency situation is much longer than the parasympathetic nervous system. So although you think that it's important for the message to arrive quick, but because it is going to be active for a long time, the speed is not so important for the postganglionic neurons. So you have the advantage of not requiring so much myelination, so you are saving in energy required for myelination, but the effect is prolonged and it's diffuse. It affects many different organs. So that's why. So are preganglionic axons like the receptors, and postganglionic axons respond to the message? No, the neurons that respond to the receptors are sensory, remember? So the sensory message now is the one that brings the impulse from the receptors. And the receptors are on the organs. Okay? So remember, preganglionic and postganglionic, they're both motor neurons. Okay, so myelination costs energy. So that's why the postganglionic neurons are not myelinated. You don't need a quick response. And you see that the postganglionic from the sympathetic nervous system is long because it's going to affect many different organs. The parasympathetic is going to be more localized. So that's why you have a much shorter postganglionic neuron. Okay, so here I'm showing how, so I told you that the autonomic nervous system has a basic function that is independent of the CNS, but the CNS can still exert influence on the autonomic nervous system. So you have the limbic system, emotion, and the other regions of the brain that send inputs to the ANS. So hypothalamus, pons, medulla, and reticular formation. So you see those areas here. So in the hypothalamus, you have the osmoregulatory center, water balance, temperature. You have eating behavior. So the hunger and the satiety center are located in the hypothalamus. So these are all basic functions that the hypothalamus can have an effect on the autonomic nervous system. Then in the pons, remember the pons, the bridge, What is located? You have the secondary respiratory center, pneumostatic and apneustic center, and also the urinary bladder control at the pons. Then in the medulla, you have the cardiac centers and the respiratory centers. So all these structures exert an effect on the autonomic nervous system. So what do they control? They control heart rate, blood pressure, temperature, and water balance. So you see that these parts of the brain of the CNS, they can exert an influence on the autonomic nervous system. Okay, so here I wanted to show you how pushing the ganglions out of the CNS, how it conserves space. So here you have a preganglionic neuron, and it can synapse with many postganglionic neurons. So let's see if this works. You're going to see here the signal. See the signal coming from the preganglionic neuron, and it's going to spread out. It's going to affect many. So it's a diverging circuit. Remember, when you have one neuron that is controlling its synapsing with many different neurons. So these are the ganglia. These are the preganglionic and these are the postganglionic. So you have the signal coming from the preganglionic and synapsing with many. So by pushing it out of the CNS, this is a big, big advantage. So you have a disadvantage is that there's less brain control. But because it's the autonomic nervous system, this is not so dependent on the CNS. So you see that these are divergent circuits where you have one neuron synapsing with many, because you have all these organs in our body that need to be regulated by the autonomic nervous system. Okay, so here now what I'm showing is the complexity of the neurotransmitters and the receptors. Okay, somatic, very simple. One motor neuron, myelinated, and acetylcholine, always excitatory. So you have a nicotinic acetylcholine receptor on the skeletal muscle. Those muscles that are under voluntary control. Now look here, the autonomic nervous system. Two motor neurons, preganglionic, myelinated. And it releases acetylcholine, and you have the nicotinic receptors at the ganglia. Then you have the postganglionic that is not myelinated. And then this postganglionic neuron can release acetylcholine or norepinephrine or epinephrine at the adrenal medulla. So the effect of these neurotransmitters is to regulate heart, smooth muscle, and glands. So the unmyelinated are metabolically more efficient. Why? Because there's no myelin. You don't have Schwann cells here. To maintain Schwann cells all along this postganglionic fiber, that would require large amounts of energy. So not having myelin here is very important in terms of conserving energy. And you don't need such a quick response for the glands, for the viscera. Okay, so the autonomic, the efferent part or the motor component of the autonomic nervous system it has two divisions, parasympathetic or sympathetic. So the parasympathetic is the one that is dedicated to maintaining homeostasis. So acts to conserve, save energy, relaxation, rest, or digestion. So during digestion, the parasympathetic nervous system is active. And that's why when after a meal, you don't want, for example, this myth that after you have a large meal, you shouldn't go swimming because there's a shock. You go into very cold water, and that's an emergency situation. So you don't want the sympathetic nervous system now to be activated. So digestion, relax. Let your body be concentrating on digestion. Now, if there is an emergency situation, fight or flight, the sympathetic nervous system comes in. And now the sympathetic nervous system activates or inhibits many different functions, many different organs. So that's why you have that very long postganglionic neuron that can synapse with and regulate many different organs. Okay, now organs, the different organs are innervated. What does that mean? They receive input from both the parasympathetic and sympathetic, and they usually have opposite effects. So, for example, the parasympathetic decreases heart rate, decreases blood pressure, increases digestion. The sympathetic increases heart rate, increases blood pressure, decreases digestion, and increases metabolism of fat and glycogen. So you have the different organs receiving input from both motor divisions, parasympathetic and sympathetic, having opposite effects. And of course, they're not going to be both active at the same time. So the activity comes from the receptors. Going back to the autonomic system, is the unmyelinated part what allows for noradrenergic to be released? Well, let's see where we have here, the noradrenergic, right? Here you have, so you have here the preganglionic, when it's active, it releases acetylcholine. You have nicotinic receptors here on the postganglionic, so it's excitatory. And then you release norepinephrine. Okay, but now the norepinephrine can be excitatory or inhibitory. So for the norepinephrine to be released, you need the preganglionic neuron to be activated, right? So this preganglionic is myelinated. It releases acetylcholine that triggers an action potential on the postganglionic, which will release noradrenaline here, or acetylcholine here, or epinephrine here. So you're asking, does the non-myelinated part affect the release of, well, the non-myelinated means that these neurons are either cholinergic or adrenergic or epinergic. Okay, the activation of these postganglionic neurons are what regulates the release of the neurotransmitters. And then these neurotransmitters are going to act on the organs and depends on what receptors are on these organs. You will soon learn this. So these organs, the heart, has both cholinergic and adrenergic receptors, and so on. So it depends on which neurotransmitter is going to be released and binds to its specific receptor on the heart, and it's either going to be the heart is going to be, the heart rate is going to increase or decrease. I hope this answers your question. But as we go along, you will understand better when I give you examples. Okay, so now it's just like a geography kind of minute where we're going to discuss where are the cell bodies of these neurons, the preganglionic and postganglionic. So first, let's look at the preganglionic. Where are the cell bodies? Okay, if we focus on the parasympathetic nervous system, so the cell bodies of the preganglionic neurons, where are they located? So parasympathetic, so they are located in the cranial and sacral areas. Cranial means cranial nerves, and all the way here at the end. So top, all the way at the top, and all the way at the bottom. So the cell bodies of the parasympathetic preganglionic neurons are either at the brain, and that's the case of the cranial nerves, or all the way at the bottom, the sacral nerves. So brain
Professor,
stem, just a minute, cranial nerves 3, 7, 9, and 10. 10 is the vagus. And then at the sacral area, S2, S3, S4. Yes, please.
sorry, I just wanted to ask these locations, cranial sacral, is this where the preganglionic neurons start? It's the point of
Yes,
origin?
exactly. Exactly, where there's cell bodies. So you have the cell bodies, right? We're talking about where are the cell bodies, okay? This is a preganglionic neuron, right? Where are these cell bodies located? So for the preganglionic, for the parasympathetic nervous system, those cell bodies are located either in the brain, and those are the cranial nerves. So cranial nerve number 3, 7, 9, and 10, or they're located all the way at the bottom, the sacral, the spinal nerves, sacral, S2, S3, S4. So those are the parasympathetic. How about the sympathetic? Where are the cell bodies of the preganglionic neurons of the sympathetic nervous system? Where are those located? Those are located in the spinal cord, the thoracic and lumbar area of the spinal cord. So parasympathetic, top and bottom, and sympathetic in the middle. So here you see again, you see here the parasympathetic, so the cell bodies are located either in the brain or all the way down at the bottom, at the sacrum. So cranial nerves 3, 7, 9, and 10, midbrain, pons, and medulla, and then all the way at the bottom. Now the sympathetic nerve roots, so the cell bodies, they're located on the spinal cord in the thoracic and lumbar area. So this is a very good illustration showing the origin of the preganglionic neurons. How is a question on ganglion going to be asked in the exam? We will see this next week. Okay, I can say, where is the origin? I don't know. Right now, I'm focusing on the material, not on the questions. Okay? Okay, I could ask the location of the cell bodies of parasympathetic or location of sympathetic, and so on and so forth. Okay. So compare the anatomy of parasympathetic and sympathetic. So these are the motor neurons. Okay, parasympathetic pathways. Preganglionic originate in the brain stem, the brain, the medulla pons, midbrain pons medulla, or sacral region of the spinal cord. The ganglia are located on or near the target organs. They have long preganglionic, so parasympathetic, long preganglionic, but short postganglionic. Sympathetic pathways. Where are the cell bodies of preganglionic? in the thoracic and lumbar region of the spinal cord. The sympathetic ganglia are located in the chains, parallel, outside the spinal cord, but parallel. They are short preganglionic that form white rami. Why is it white? Because they are myelinated. That's why they give an appearance of being white. The long are postganglionic. They are not myelinated, so we call them the gray rami. White rami are myelinated, gray rami are unmyelinated. So here again, you see the differences. Points of origin, sympathetic, T1, thoracic, lumbar. Here, parasympathetic, brainstem and sacrum. Site of peripheral ganglia. Anyway, this is repetition. Okay, so now let's see. We saw where the origins of the preganglionic. Now let's look at what happens to postganglionic. Okay, this side is parasympathetic, this side is sympathetic. But remember, this occurs on both sides of our body. This is just for simplicity. So preganglionic, very short, sympathetic, very short, preganglionic, myelinated. They synapse in this chain of sympathetic ganglia. So it's a parallel formation of ganglia that's parallel to the spinal cord. So very short preganglionic, synapse at the ganglia chain, and then the postganglionic, very long. And they innervate pupils, saliva, heartbeat, bronchi. They regulate the movement along the digestive tract, liver, adrenal gland, and bladder. So you see that you have these very long postganglionic neurons. On this side, parasympathetic. The preganglionic, the cell bodies are at the brain, at the medulla pons and midbrain, and all the way sacrum. And you see that the preganglionic are very long, and they synapse very close to the target organ. Now, notice here, very, very important aspect of the parasympathetic. 90% of the parasympathetic innervation is carried by the vagus nerve. So one cranial nerve number 10, the vagus, controls all these organs, the heart, the lungs, the movement along the digestive tract, and the liver. All this is regulated by the vagus nerve, which is a parasympathetic postganglionic. It's not, it's a preganglionic, right? It's a preganglionic neuron from the parasympathetic nervous system. Very interesting that all these organs are regulated by one parasympathetic neuron. Okay, sympathetic. So now, I don't know if I want to go through this. It's just very quickly. So here you have the ganglia, showing the ganglia on both sides, the sympathetic, the chain of sympathetic ganglia on each side of the spinal cord. Some are collateral. You have some additional ganglia that are collateral. That means that they are outside of the chain. And then you see the long sympathetic postganglionic neurons that are innervating eyes, salivary glands, and so on and so forth. And here's the adrenal medulla. So this is sort of a view, complex view of the sympathetic nervous system, where the preganglionic, the cell bodies, are in the spinal cord, thoracic and lumbar, and go out to the periphery. Now, the adrenal gland. You see here the kidneys, and above the kidneys are these glands known as the adrenal glands. And the adrenal glands have a cortex. So this is a cut through the middle of the adrenal gland. And you see that it has a cortex, and inside it has a medulla. The cortex, the outside of the gland, produces hormones, glucocorticoids such as cortisol, mineral corticoids like aldosterone. It's very important in regulating salts and water, and androgens. So it produces testosterone. So women have testosterone because it's produced in the cortex of the adrenal medulla. But now the middle, the adrenal medulla, that is like a neuron. It's like a modified post-ganglionic neuron, and it releases epinephrine. And it plays a major role in the heart, control of the heart rate and blood vessels. So here you see the spinal cord. You see a long preganglionic neuron. This is sympathetic, and it goes right to the medulla of the adrenal gland, where it releases acetylcholine. And here you have a very small postganglionic, it's a modified postganglionic sympathetic neuron that releases epinephrine. Epinephrine is released into the blood and travels through the blood vessels, and it reaches the heart. So here you have the adrenal medulla. It secretes norepinephrine and epinephrine. It's a modified sympathetic ganglion. It receives input, preganglionic input from the spinal cord, and it releases neurohormones into the blood. Okay, now if you concentrate on this superior cervical sympathetic ganglia, so we go here, we see the superior sympathetic ganglia. So it sends input into the eyes and the eyelids. So what happens here, if there's damage to that superior cervical ganglia, then there's a problem with the eye, drooping or weakness of the eyelid. It's known as ptosis. And the pupil, the side where there's the damage of the superior cervical sympathetic ganglia, what does that affect? Affects the eyelid, affects the pupil. You see the size, the different size of the pupil on the size where the ganglia is damaged. And also it affects the perspiration. So this superior cervical ganglia controls the eyelid, the pupil, and perspiration or sweating. And this is known as the Horner's syndrome. Is this why vagus is the longest cranial nerve? Yes. And there's a lot of studies that relate the vagus nerve, because the vagus nerve is so large, and think if there is retrograde transport, right? So it goes right from here to the brain. So a lot of the digestive tract, so we have many bacteria, flora in the digestive tract. So their chemicals can be conducted via retrograde transport all the way to the brain. So there's a lot of research going on, making the connection between the vagus nerve and the functioning and degeneration of the brain. Okay, so here you have a view of the parasympathetic nervous system, where you have long preganglionic neurons, the cell bodies located midbrain, pons, and medulla, and at the sacrum. And here is the vagus nerve. Look at this, how it forms these complexes, these plexuses that will innervate all these different organs. So they are controlled by the vagus. 90% of the parasympathetic outflow is carried by the vagus nerve. Okay, so this just compares what I've been saying, the architecture of these two divisions, motor divisions. Now here we go on to the neurotransmitters and receptors. So here, sympathetic, you have preganglionic releasing acetylcholine. Here you have nicotinic receptors that are excitatory. This is the postganglionic. So when it's activated, then you have the action potential releasing norepinephrine that will bind to adrenergic receptors on the target cells, or acetylcholine receptors on salivary glands. Here, the parasympathetic, you have acetylcholine and nicotinic receptors, just this is common to both. And here you will always have acetylcholine. So the one that is different is for the sympathetic nervous system. Okay, so enough about the geography and receptors and neurotransmitters. Now let's move on to the enteric nervous system. So there's a whole nervous system that has more neurons than the spinal cord, and it's dedicated to the digestive tract. So here you see a cross section of the digestive tract. So let's say you go through from the esophagus all the way to the anus. You have this enteric nervous system, and it's arranged in two layers. One is the myenteric or myenteric plexus. The other one is the submucosal plexus. So you have these complexes going all the way from the esophagus all the way to the anus. So the myenteric plexus, where is it located? So it's a layer of nervous system that is located between the circular muscles and the longitudinal smooth muscles, right? This is all smooth muscles. And you see here, this layer represents the myenteric plexus. Here, the myenteric plexus, located between the layer of circular muscle and longitudinal muscle. And what is the function of the myenteric plexus? The function of the myenteric plexus, it regulates the movement along the digestive tract. The submucosal plexus that is located, you see here, in the submucosal muscle, here you see, its function is to regulate the release of hormones and the blood vessels and so on. So the submucosal is mostly regulating the function of the digestive tract, while the myenteric plexus regulates the movement along the digestive tract. So the submucosal, so the myenteric plexus is dedicated to motility, moving the food, the bolus, along the digestive tract. The submucosal is dedicated to regulating secretion, absorption, and blood flow. So this is very, very complex nervous system. And you see here different views where you have the longitudinal and circular smooth muscle fibers, and in the middle you have the myenteric plexus. And then here, below the circular layer, you have the submucosal plexus. Here you see the myenteric plexus controls the movement down the digestive tract. So you know that it always goes down. It can never go up. It only goes in one direction. And the submucosal regulates secretion, absorption, and blood flow. And there's a very, very interesting TED Talk. It's 15 minutes. And if you are in the mood to listen to it, just go to this website. Just click here. It's very, very fun 15 minutes to listen to. Okay. Now, sympathetic nervous system. You would think it's activated in an emergent situation. So you might think, oh, this is going to be on and off very quickly. No, because it's an emergency situation. It's long lasting. You have to affect many different organs. And it's time-wise, it's long lasting. The parasympathetic is not so long. Its activity is to conserve and restore body energy. So it's involved in homeostasis. So under physical stress, sympathetic nervous system is the dominant. Okay, now you know that the neurotransmitters, at the end of the postganglionic neuron, you have the varicosities. And the varicosities house the synaptic vesicles with the neurotransmitters. And now you know these neurotransmitters can be norepinephrine or acetylcholine. And they are released from these varicosities. So we're going way back to the beginning of this set of lectures. Now here you see the postganglionic neuron with these varicosities that innervate the smooth muscle layer below them. So what happens if acetylcholine or norepinephrine is released? What happens? You have the action potential coming down the motor neuron, the postganglionic motor neuron, opens voltage calcium channels. Calcium floods the varicosities, so you have a lot of calcium coming in. And this allows for the synaptic vesicles to release the neurotransmitter by exocytosis. So the more neurotransmitter that is released, the stronger the response. Okay, and I already talked about this, how you have different neurotransmitters and different receptors that are being released. Okay, how are these neurotransmitters removed? You know this already. Norepinephrine is a catecholamine. So what happens? It's released, but then it goes back inside the presynaptic or the preganglionic or actually it goes back into the varicosity of the postganglionic neuron, right? You have transporters that bring it inside where it is metabolized by monoamine oxidase or COMT. It can also diffuse out to the blood vessels. So it depends on these transporters, depend on the transporters to remove the neurotransmitter from this synapse. When it's acetylcholine, you have the acetylcholinesterase. So the same thing happens here in the autonomic nervous system, the way that you get rid of these neurotransmitters. Except, remember, that you don't have astrocytes. In the brain, astrocytes can remove some of these neurotransmitters, like glutamate. Okay, now there's going to be a higher level of complexity. So we have the different receptors, right? We have nicotinic receptors that are located at the ganglia, at the cell at the postganglionic neurons. So here you have always nicotinic receptors. But at the postganglionic, you can have muscarinic receptors or adrenergic receptors. So here in the parasympathetic, on the effectors, it is going to be always muscarinic receptors. For the sympathetic, you have noradrenergic or muscarinic. Now, there are different types of muscarinic and noradrenergic receptors. Some are alpha, some are beta. And that's why sometimes you can have activation or you can have inhibition, because the different types of receptors can be inhibitory or excitatory. So now we have a different type of complexity. So we have different neurotransmitters. We have different types of receptors. And now within one type of receptor, some can be inhibitory, some can be excitatory. Okay, so here you have the sympathetic, preganglionic acetylcholine, both in sympathetic and parasympathetic. So this lecture is a little bit dense, but it's very important to know the receptors. Why? Because, when we're sick, we have our different viscera that are not working properly. They have different receptors. So if we know exactly what receptors are present, then we can design drugs that are more specific for one receptor and not another, and therefore decrease the side effects. So that's why it's so important to know all this knowledge. And there's so many funds invested in studying these receptors and designing drugs that become more and more and more specific. So, when we talk about postganglionic, at the sympathetic, we have adrenergic receptors. At the sweat glands, we have muscarinic receptors. In the parasympathetic, it's cholinergic and they're muscarinic. In the sympathetic, they can be alpha or beta. Okay, so now let's go to the next level of complexity. So adrenergic receptors. Adrenergic receptors bind norepinephrine or epinephrine. Okay, so there's alpha and beta. So alpha 1 and beta 1, they're usually excitatory. So these are proteins, remember, they're proteins. They're specific genes that encode for these receptors. So alpha 1, beta 1 are excitatory. Alpha 2, beta 2 are inhibitory. Beta 3 plays an effect in thermogenesis, but there are exceptions. So, alpha 1 receptor, it's excitatory. Where is it located? On smooth muscle cells. So when it's activated, it causes contraction of the blood vessels. So activation of alpha 1 will induce contraction of the blood vessels. When you need contraction of the blood vessels, remember this is a response, for example, to cold temperature. You need the blood vessel diameter to shrink. So this is because of the alpha 1 receptor is being activated. Alpha 2 receptor is inhibitory. And so when it's activated, it causes relaxation. So it's opposite of alpha 1. So it's located in smooth muscle, so it causes dilation of the blood vessels. Please don't make the test as complex as these neurotransmitters. Mohammed, you have to have a notion. It's just for a short term. I'm going to tell you what you need to know, okay? So now we go to the beta receptors. Beta receptors, beta 1 is excitatory. So it stimulates heartbeat. Beta 2 is inhibitory, causes relaxation, and beta 3 is involved in heat production. So I'm going to give you sort of an example that makes this all more interesting and less complex. So here you know the nicotinic receptor is ionotropic, the muscarinic receptor is metabotropic, and you know this already. I don't want to go over it anymore. And here you have alpha 1, alpha 2, beta 1, beta 2, and its effects. So what I want to reach here, this now puts this in context. So let's think about the heartbeat, okay? On this side, it controls emergency situations, sympathetic. On this side, it controls relaxed situation, parasympathetic. So let's say there's an emergency situation. Your heartbeat starts pumping much, much faster. Why? So here you have the blue neurons being activated. So we're talking about the postganglionic. So the postganglionic is going to release norepinephrine at the heart. It's going to activate beta 1 receptor. Beta 1, heart muscle, increases heartbeat. So you have norepinephrine activating beta 1. You see the heart pumping. Okay, so when your heart starts beating very, very fast, remember, it's the norepinephrine, it's the sympathetic, postganglionic norepinephrine binding to beta 1 receptor. Now you need to go back to relaxed stage. Now you have the parasympathetic here in pink, and it releases acetylcholine. It inhibits, it's inhibitory, so it decreases heart rate. So you see here the situation where you need the heartbeat to increase under emergency situations. You have sympathetic, norepinephrine, beta 1 receptor. And here, when you need the heartbeat to relax, you have as parasympathetic is activated. The same for the pupil. So under emergency situations, you have sympathetic, you have norepinephrine being released and causes the activation of the radial muscles. So you have pupil dilation. Then going back to relaxed stage, you have the parasympathetic being released, acetylcholine, and the circular muscles are going to be activated, bringing the pupil, shrinking the diameter of the pupil. Okay, so all I want you to know is this chart. So I could ask you, how can you induce dilation of pupils? And you would say the sympathetic nervous system is activated and so on. So this would be a nice chart for you to remember. Okay, now for example, one advantage of knowing all of these, oh my gosh, it's so late. So for controlling the blood pressure, so maybe I will just go through this and then I will finish it on Thursday. So the beta blockers, so decreased blood pressure. So one of the first medicines that was developed to decrease blood pressure, that's very important, right? To decrease blood pressure. This drug, propranolol, it binds to all different types of beta receptors, of beta adrenergic receptors. So it has huge side effects. So it decreases blood pressure, which is what we want. It decreases heart rate. But what are the side effects? Hypoglycemia and narrowing of airways. So out of breath, that's a huge side effect that you don't want. So by knowing what receptor is important, so this other drug, metoprolol, was developed at a specific beta 1 blocker, so it doesn't have these side effects. So I know it's very confusing and you have all these neurotransmitters and receptors and so on. But my idea is to give you the notion of how complex this is and how can we adapt to all these situations and how things can go wrong and how important it is. When you go to the doctor and you get a medicine, you should know what is the target, what is the receptor that is going to be affected. And for today, this is enough. And I will see you Tuesday. And Tuesday will be very few slides. That's it, 3 slides. And it will be review. I will have lots of questions. So review on Thursday. And good luck on your lab exams. Okay. Oh, okay. So that's good. You see, Heather, now you know. Buddy, see you on Tuesday. Don't forget to look for your grades. Make sure that they are all correct. Okay, bye bye, everybody.